Okay, hello everyone. I'm very warm welcome to another webinar from A&T Neuro Educational Webinar Series. My name is Farnoosh. I'm the head of education, and very happy to be hosting today's session, which is about fundamentals of topographic EEG analysis. Our presenter today is Professor Micah, who trained at the Johns Hopkins University and then Albert Einstein College of Medicine. He is the founding director of the Laboratory for Investigative Neurophysiology, and he is also the director of the Sensory, Perceptual, and Cognitive Neuroscience section of the Center for Biomedical Imaging. So let's get started. Micah, thanks for being with us. The screen and the stage is yours. Please go ahead. I will just switch the screen. We can raise the poll questions whenever you want. I just turn off my webcam. Thank you.
Right. So hello to everyone out there. It's very unusual to give this type of presentation where I can effectively only see my own screen, and I'm very grateful that so many of you have signed up and have shown an interest in what I have to say today. Hopefully I won't disappoint, and we'll have a nice lively discussion. Please do indeed send me any questions that you may have offline. Let's see if I can get this proper. Presumably now you can all see my slides well. Let me do this again so that you're not seeing me. There we are. Right, so today what I'm going to do is give you a bit of an overview of some fundamentals of topographic EEG analyses. And it's mostly targeted today towards newcomers and those who are perhaps unfamiliar with why topographic analyses are important and informative. For those that are more aficionados, I beg your patience, but by all means contribute to the chat. So here we go. The most important thing is to remember that none of us are working alone. Even in these times of COVID-19, it takes a team of individuals, and I immediately want to thank those that I've worked with either in the past or currently work with on developing some of these techniques and approaches for EEG analyses. This includes Christophe Michel, first and foremost, who convinced me to come to Switzerland, and together with him and Denis Brunet, we've worked very hard on a software called CarTool, as well as some of the folks in the lab that have now gone on to greener pastures, including Marcia, Atina, and Jean-Francois, as well as some of the current members in the lab, Andrea, Benedetta, Roxandra, and Chris. So I think it's important to always start from a theoretical standpoint of what we want to study, what we want to understand about the human brain. And I think William James put it quite well to suggest that there's a part of what we perceive that comes through our senses from the object in front of us, and another part, and it might be the larger part, that always comes out of our own mind. Now, this was back, of course, in the late 1800s, but we can put this in more contemporary terms by thinking about how sensory information can shape brain activity, but also how another portion that is ongoing brain activity or past experiences, our intentions, so on and so forth, can shade and shape brain activity. And this, of course, can happen over time. So how do we access this? How do we get into a question of who speaks to whom, when, say what, and to what end in the brain? So these are, I think, the fundamental questions that occupy most of our research activities or clinical activities. And to do that, we've been very fortunate to be around in a time where we have non-invasive techniques to investigate brain function and brain dysfunction. But I think it's also important to recall that these methods are young, relatively speaking. EEG is, of course, the oldest of these, that was introduced around 1924, followed by MEG in the late 60s, PET in the mid 70s, TMS in the mid 80s, fMRI in 1990, and FNIRS in 1993. Naturally, it's also important to bear in mind that any technique that we're going to use has strengths and weaknesses. It has the ability to resolve things either in time, in space, as well as on an axis in terms of causal inference or correlational inference. And it's important to bear all of these aspects in mind. I don't have the time nor the expertise to go through all of the techniques today, but we'll stick a bit with EEG and in particular event-related potentials as an example of EEG analysis. So the first and foremost thing to remember is what we're measuring. EEG is a measure of neural activity. It's recorded from the scalp, and we're not measuring action potentials. Action potentials, of course, are single unit recordings from individual neurons. It's at a much finer spatial scale than we can resolve with EEG. And we're not really measuring local field potentials either. Instead, we're measuring population responses of postsynaptic potentials. That is, anytime there's a postsynaptic potential, there's, of course, an extracellular current sink and a current source, which can then be modeled as a current dipole. And if this is strong enough and synchronous enough across space, then it can propagate to the scalp. So it's only a certain variety of neural activity that we're actually able to detect with EEG. So it's very important to bear in mind, of course, what it is that we're measuring. So first and foremost, as a poll question, so now is your chance to take part. I'll put to you the following question: when was high density, that is 64 channel or greater EEG introduced? Was it the 1950s, the 1960s, the 1970s, 80s, or 90s? So I'm not quite sure how long I have to give people to reply, but I'll let Farnoosh organize that.
Let's give this another 10 seconds.
Okay.
So far, 55% voted. Let's wait for the rest.
Don't be shy. Take a guess if you don't know.
Okay, let's share the results. Do you agree with that, Micah?
Yep, that'd be great.
Perfect. Please go ahead.
So to do the answer is in the 1970s, but I don't know if people got that right because I can't see the poll results. But I'll presume that you guys did all right. I'm not sure if I'm controlling things properly from my end, but I'm going to move on, and hopefully you guys got that one right. So indeed, EEG has been around since the late 1920s, and it's been developed not only from a technical standpoint, but also in terms of findings and basic findings and applications. So the 1970s, in particular, Derek Fender out at Caltech was developing some of the first high-density EEG caps, which you can see here in this excellent photo from Caltech magazine back in, I think, 1972, if I remember properly. And you can see it's moved a long way from pen and paper EEG up through some of the great 1980s images of what EEG caps can look like, into more mobile devices. Here is an ANT system, as well as completely mobile and wireless EEG that can be used in all sorts of environments. We'll come back to that momentarily. So let's think first a little bit about some general things about EEG in terms of cost-benefit analysis. I would put to you that EEG is quite cost-effective. It may not seem like it, but EEG is actually relatively inexpensive. So it's on the order of about 60,000 euros for a 128-channel system. The other good thing out there is that there's a number of freeware and open source software packages for handling data, which makes it highly cost-effective in my opinion. The long lifetime of EEG hardware makes it possible to invest once and have it last a good long while. And that makes the lifetime of the equipment quite long, and therefore quite cost effective again as well. It's portable. Systems now are very lightweight and even wireless. They're easy to use. It's easy to train up medical staff, students, technicians. You don't actually need a technical expertise in terms of from a medical standpoint for most research applications. The temporal resolution, of course, is on par with neural activity, that is sub-millisecond, and you can record simultaneously from a large number of channels. EEG is accessible. You can record from neonates to elderly, people with claustrophobia, implants, pacemakers, clinical populations, you name it, really. It's hard to imagine a population that we can't really access with EEG. And it's an entirely versatile technique, meaning it can be combined with other techniques, whether it be things like fMRI, MEG, PET, FNIRS, non-invasive brain stimulation. It can also be combined with pharmacology and physiology. And because it's portable, it can be applied in real-world environments. So it's also important to think about how EEG can be used independently of overt behavior. Individuals can even be anesthetized, and you can still be looking at sensory, perceptual, and cognitive functions. So time for the next poll question. So get ready. MEG has a higher spatial resolution than EEG. True, false, or I love fMRI?
You have 30 seconds for this.
And you can only give one answer.
Okay, let's see the results. So 66% said true, 28% false, and 7% I love fMRI.
It's good to see everybody has a good sense of humor out there. So the correct answer is false. It's commonly thought that MEG actually has a higher spatial resolution, but that's actually not objectively true. Undeniably, fMRI has a higher spatial resolution, but simulations, empirical work that have compared the localization error across methods show that EEG can have a localization error below 1 centimeter. People have also directly compared MEG and EEG performance using the same number of sensors, and that's an important aspect when making comparisons between the two techniques. The other important thing to remember is that EEG is sensitive to both radial and tangential sources, whereas MEG is sensitive only to radial sources. And EEG is sensitive to both superficial and deep sources. So these are actual strengths of EEG over some other techniques. It's not to say that people shouldn't also do MEG or fMRI research. I really don't want to give the impression that people shouldn't be doing other kinds of research. However, some of the common beliefs out there about EEG might need some updating. And people should bear in mind that the localization precision of EEG is really quite high, and in fact, can outperform MEG. The other common belief is that EEG has a low signal-to-noise ratio, and this is not true either. Evoked activity is visible in single trial data. These are just two examples where we've specifically addressed this point. There's also some work by Charles Schroeder and his colleagues also looking at this not only in humans, but also in non-human primate models. There's also a remaining issue, in my opinion, about a weak analysis pathway. I think this is true. I think some of the open source and freeware initiatives are changing this, as well as a general renaissance in the use of EEG. This isn't helped by some of the, what I like to think of as a bit of an alphabet soup when it comes to components being associated with given cognitive processes. People get a bit confused and lost as to what am I looking at and what do I want to do. And of course, there's a large and diverse profile of the users of EEG with different research agendas and, quite frankly, a lack of stringent guidelines for the use of EEG, both in research and clinical practice. I think the other major weakness at the moment is that we despite the long history of EEG, in many ways we have a weak understanding of the underlying neurophysiology, and more often than not, a weak understanding of the interpretability of the data. And that will be the focus of the rest of the talk, but we'll get to that in a moment. I believe in general, there's been a split between those people that want a correlate of a presumed brain process and those that are really looking to understand the underlying neurophysiologic mechanisms. This leads to many impediments, I think, for some of the data interpretations out there. I think there aren't enough studies about comparative neurophysiology. In that regard, I think predominantly of people like Herbert von Jr. And again, Charles Schroeder, both of whom were working at Albert Einstein College of Medicine. I had the good fortune to work with both of them. But what they did was compared intracranial work in non-human primates with surface recordings in non-human primates, which allowed for some important insights about the underlying neurophysiology of EEG. And of course, biophysics can help us look at some of the underlying mechanisms more intensively. So if you think about a typical analysis pathway, we often start with some type of experimental protocol. We acquire data, we might re-reference data, we might filter the data, remove artifacts, epoch the data, and then we can either go into a straightforward signal averaging and statistical analysis, or we can start moving into more complicated analysis approaches, which are currently quite fashionable, based on classification techniques, decoding techniques, and even brain-computer interfaces that allow us to close the loop and go back and impact the research protocol that we're working on. So these types of analysis pathways are all there, but how do newcomers and how do people perhaps less familiar with EEG enter into this either for the first time or to understand what's going on in the literature? So to do that, let's take a step back and perform a Gedanken experiment where we may be an experimenter, depicted by this very fine gentleman here, and we want to test this other fine gentleman and know whether or not he's telling the truth. Is he sick? Is he well? Does his brain treat information one way or another way? You can imagine any type of research question that you might have. And to get at this question about truth, is he telling the truth? In the film, of course, Meet the Parents, Robert De Niro's character is using a polygraph machine, but this might as well be an EEG machine. And we can attach it to our participant and want to know whether or not Ben Stiller, in this case, is telling the truth about his intentions with one of Robert De Niro's daughters. So this is a very typical EEG setting, and what we want to know is what in our data is truthful, what is objectively going on in a given experiment. And any number of different books in the literature will tell you what to do. And this is just a sampling of some of the books that are out there about EEG analyses. And I'll single out one of them in particular because it's become quite popular over the years. Steve Luck's very, very accessible book talks about a phenomenon that he terms the no Switzerland principle. And being a good Swiss American, this caught my attention. And it is based on the fact that there is no electrically neutral site on the head or body. This is biophysics. So therefore, any electrical measure of voltage, which is measuring potential between 2 points, is the difference in voltage between two sites. And that's an inescapable truth about measuring voltage potential. So therefore, you always need a reference site in order to do your recordings. So that's important to bear in mind, but what's also important to bear in mind are what are the consequences of this? What does it mean to our measurements, to our interpretation of data, if this no Switzerland principle is indeed an undeniable truth about the biophysics of EEG? And now I'll put to you another poll question, which is the choice of reference just redistributes my results. It doesn't really have an impact. All it does is move around where I see my results. So is this statement true, false, or do you only do replication studies anyway? So again, we'll give you, I think, 30 seconds or so to to reply to this?
Yes, the poll is ongoing. Around 50% of the people voted. Let's wait for the rest. Perfect. I close the poll, and there are the results. So true, 32%, false, 64%, and I only do replication studies 5%. Fair enough.
Fair enough. Well done, everybody. Indeed, the choice of reference does not just redistribute your results. It can dramatically alter your results and therefore your interpretation. And let me walk you through why that's the case. So imagine that you have two different stimuli, let's say Steve Luck's book and Christophe Michel's electrical neuroimaging book, and you want to know how the brain responds differently to these two stimuli. And you have three electrodes that you put on the scalp. You measure event-related potentials to these stimuli, and you choose a reference among your three electrodes, and you find no reliable differences. Now, you might say, well, I have fewer electrodes, so there's fewer chances for me to make a mistake in my experiment and in the interpretation of my results. But a colleague of yours might come along and say, you know what, I'm going to repeat your experiment. I like replication studies. And I'm not only going to repeat your experiment, I'm going to collect data from 128 channel EEG. And I'm going to do the same analysis that you did. And when I do, I get no results. But because I took a basic statistics class, I might think, well, I have more electrodes, so do I have more chances for error? And when I then take a different reference, because I have all of these electrodes on the cap, in my electrode cap, I can choose a different reference. And when I do, lo and behold, I get a nice whopping effect between these two stimuli. So now it begs the question, who's right? Is there a difference between these stimuli, or isn't there a difference between these stimuli? And we can view these types of analyses another way. If we were to conduct a mass univariate analysis across every single electrode as a function of time, setting some threshold for what we accept as a statistically robust effect, which isn't the topic of conversation today, but we can certainly discuss it. One might imagine seeing a result, let's say a main effect of the two stimulus conditions around 150 milliseconds, and maybe another effect around 300 milliseconds, using a given reference. But if we change the reference, doing the same analysis, what we observe is maybe in this case there's still an effect at 150 milliseconds, but it's not a main effect anymore. It's now an interaction between scalp location and condition, which would change our interpretation entirely of the outcome. And we can see this later effect gets much more robust, statistically speaking. So again, going back, going forward. We can look at this more quantitatively in another example. Here is a paradigm where we had initial and repeated presentations of the same stimuli, looking for repetition suppression. So you're seeing the initial presentation in black, the repeated presentation in red, the difference between them in blue, and in green zones in time when there was a robust effect, that is p less than 0.05 for at least 20 consecutive milliseconds. On the right side, you're seeing the scalp topography of these voltage potentials. For the initial presentation, repeated the difference as a function of time. And in the bottom, you're seeing which electrodes at that given moment in time are exhibiting a statistically robust difference. So this is with a vertex reference. If we change the reference to an average mastoid reference, you can see the effects change quite dramatically. So again, here, the event-related potentials look quite large. And the effects, let's say, starting around 250 milliseconds or so, affects, statistically speaking, predominantly over the posterior scalp. Whereas if you change the reference, not only do the waveforms change, no difference now at this electrode P6, But all of the statistical difference not only moves in space, but also moves in time. So things can look very, very different depending on what reference choice one makes. So you're in a bit of a quandary. Is there an added value in high density EEG? So again, I'll put this to you to think about, at least for the next 30 seconds or so, and we'll see what you all think. Okay.
I close the poll. Let's share the results. 88% true, 5% false, and 7% I just want my work published.
Again, good to see the audience has a good sense of humor in there. Indeed, there is added value, I would argue, in high density EEG. But we'll come back to this. It's an important question. But for now, I'll agree that indeed there is added value. But what is that added value? So one can imagine two sets of advice. Some might tell you to record just from 16 to 32 electrodes, analyze single values after averaging across relevant scalp regions or components. You can report the interactions that you might see between electrode and condition, but then don't say too much about it. Whereas others might encourage you to record from at least 64 channels, analyze reference independent and global features of the brain's electric fields of the scalp, and to report these modulations in strength and topography independently. So this is the advice out there, but what does that mean? How do you actually do this? So this might tempt one to think that there are added headaches in high density EEG. Now, again, poll question to you, is this true? Is this false or not tonight, honey? Okay.
Okay, nearly 70% voted. Let's see the results. 53% the first option, 32% second option, 16% the third one.
Wow. All right. Well, we can see the effects of isolation on people's moods. Very good. So yeah, there may be some added headaches, but I'm going to try and demystify that. And the main goal of today's webinar for me is to try and take away maybe some of the mental obstacles for looking at high density EEG in a much more data-driven way and what I would argue a much more interpretable manner. But bear in mind that there are probably some peer pressure problems that add to your headaches. And it should be obvious to people that analyses that somebody suggests aren't necessarily better just because somebody of prominence advocates them or because somebody says this is the way to do it. Instead, I would say that analyses should strive to minimize the experimental bias and should be replicable and hopefully unequivocal. This is particularly important in clinical research because it can mean the difference between life and death. It can mean the difference between continuing care or discontinuing care. It can mean the difference between deciding a treatment is effective or not, and so on and so forth. So for any of you doing clinical research, and for any of you doing basic research, this aspect of replicability and objectivity, I think, can't be stressed enough. So that led me to think about proposing a thing called the Iowa Principle, which I think of as the consequences of the no Switzerland principle. For those of you that don't know the state of Iowa, it's very beautiful, but it's very, very flat compared to Switzerland, which of course has lots and lots of mountains. So I would put to you that voltage waveforms are reference dependent, which we've already seen earlier in the talk. Therefore, waveforms provide very limited neurophysiologic interpretability. And I'll explain what I mean by that momentarily. And because of this, voltage waveforms are inherently ambiguous, and they can get one in the middle of nowhere. And I think this is, if nothing else, this would probably be the key slide to remember of today's presentation. So can we surmount this? Can we get around the Iowa principle? Well, either yes, no, or what's in Iowa? You already know the answer to what's in Iowa. It's a beautiful state in the middle of America. So hopefully you won't choose that one, but let's see if people are just going to be a bit snarky. Okay, hopefully everybody's had enough time to answer.
Let's see the results. 73% yes, 10% no, 17% what's in Iowa.
Excellent. Well done, everybody. All right. So how do we get around the Iowa principle? And for that, I think it's also useful to go back and think about what do we really want to know from analyses of EEG, or in this case, event-related potentials. Well, we want to know, do we have a change in amplitude? Do we have a change in strength of the brain activity or between populations? Do we have a change in the network of brain activity? Do we have a change in the latency of brain activity? Can these be disentangled from one another? And can we achieve this in individual brains? And I would say that high density EEG can help us gain this. And showing here a view from not so far from my house of some mountains called the Dent de Midi. And the castle here is the Chateau Chillon, where Byron wrote The Prisoner of Chillon many years ago. So how do we gain this benefit of high density EEG, and how do we get around those headaches that we all want to avoid? Well, I would again put another principle to you that I would suggest is the Swiss Alps Principle. And this was very much inspired by Dietrich Lehmann, who worked in Zurich for a very long time, who was really one of the pioneers of spatial analysis or topographic analysis of EEG. And he often got people to think about mountains and the Alps. And he tried to remind people that topography is reference independent. And I'll show you what I mean by that exactly in a moment. Topography is also therefore interpretable neurophysiologically. Helmholtz already described this back in the mid-late 1800s. If you have a change of topography, it's a change of the network active in the brain. And therefore, it's unambiguous about what's driving a change in topography. So let me show you exactly what I mean. You already got a hint of that in some of the prior slides. If I were to look at the topography of EEG or an event-related potential, what you observe if you change the reference is that the colors change, but the actual shape of the topography, the three-dimensional configuration over space, doesn't change. You're simply moving sea level, if you will, around. So again, Dietrich Lehmann was very good at describing this to people. If you were a geographer making a topographic map of the Alps, the shape of that mountain, how hard it would be to walk up to the top of a mountain, wouldn't change depending on where you drew sea level. So the steepness and flatness, the topography, is reference independent. And since we know that changes of topography equal to change in the network, being able to detect topographic changes is therefore directly neurophysiologically interpretable and quite informative about the processes that might be underlying our experiment. Okay, so this is all theory right now. But how do we quantify this? How do we do this in a headache-free way to quantify our data, take advantage of high-density EEG, and have a stronger and more data-driven and therefore less biased interpretation of our data? So the way that you can do this, the way you can measure hilliness or alpness, if you will, is simply using a measure called global field power, which is not mathematically complicated. It's the spatial standard deviation. So the global field power is the root mean squared of all of the potential values at each electrode. And this is very similar to the formulation for standard deviation. So if you're comfortable with calculating standard deviation, you should be very comfortable calculating global field power. Now, what global field power does is it tells you how hilly is your topography. How hilly is your event-related potential, or your EEG, at a given instant in time. What it doesn't tell you is whether or not those hills are the same. So the global field power here and the global field power here would be identical, even though the two topographies have been turned in space, right? So it's the same thing if you were to take standard deviation of any measurement, I don't know, height of individuals in a room. It doesn't matter where the individuals are in space, you'll get the same standard deviation. And that's a shortcoming of global field power. It just tells you how strong is, or how hilly is the potential at that moment in time, but it doesn't tell you where the hills are on the topography. To do that, we have to use a different measure, which is global dissimilarity, which is exactly the same as spatial correlation. And global dissimilarity is simply the root mean squared difference between strength normalized data, or strength normalized vectors. And it's exactly equivalent to spatial correlation using this formula. So it's 2 minus dissimilarity squared over 2. So if you're used to thinking about correlation and Pearson's correlation, think about dissimilarity is not all that different, and mathematically is directly related to it. So again, to help demystify some of this, let me give you the actual maths behind this. Again, it's really not targeted today towards real experts in the field who are used to dealing with these kinds of measurements, but more for newcomers or people who are trying to wrap their head around what are the techniques out there and how do I access them. So imagine that you had a data matrix that looks like the following. You had 16 channels. Again, that's just for today's purposes. I would strongly encourage people to go to high density EEG. But today, let's imagine you only had 16 channels. You had one condition, and again, positive values will be warmer colors, 0 will be white, and negative values will be cooler colors. And you want to compare that to a second condition that is exactly 4 times stronger at each and every electrode. So I've taken the value at each electrode and simply multiplied it by 4. You can also imagine a third condition where I've inverted the value. So I've multiplied each electrode by -1. And then a fourth condition where I took condition number 2 and I've kind of moved around the electrodes in space. So I've kind of changed their distribution in space. So let's calculate now global field power for each of these conditions. So to do that, what I need to do is at each electrode take the squared value, add them up, and then divide it by the number of channels, 16, and then take the square root. So again, the maths aren't complicated. I tried to pick numbers that work out quite well and easily for today's purposes. And conditions 1 and 3, what you'll find is that the sum of all of the squared values is 64. 64 divided by 16, of course, is 4. The square root of 4 is 2. So for both condition 1 and condition 3, the global field power is identical. All the numbers are the same. Their distribution in space is different. In the same way, conditions 2 and 4 have exactly the same global field power as well, which is 8, which is exactly 4 times stronger than those of conditions 1 and 3. And again, the sum of all the values here would be 1,024 divided by 16 is 64. The square root of that is 8. So now what we see is that global field power is a measure that's independent of the spatial distribution of the data. We can now normalize our original data by the global field power. And all that means is that you're taking the measurement at each and every electrode and dividing it by its instantaneous global field power. And when you do that, 3 divided by 2, of course, is 1.5, and so on and so forth. Now we can start comparing these global field power normalized data to look for changes in topography between our conditions. So let's see how that works. to calculate dissimilarity? Well, if we want to compare conditions 1 and 2, we would take the difference between each electrode. The difference between conditions at each electrode. So 1.5 minus 1.5 is 0, so on and so forth. But you can already see that from these normalized topographies that they're exactly the same. So the sum of this, of course, is 0. 0 divided by 16 will still be 0. The square root of 0 is 0. So the dissimilarity is 0, and the Pearson correlation would be 1. These are exactly correlated with one another. If we were to do the same thing now, either between condition 1 and 3 or between condition 2 and 3, what we start getting is the other extreme. These are inverted versus each other. The sum of these difference squared is 64. 64 divided by 16 is 4. The square root of that is 2, and the spatial correlation would be -1. That is, they're inverted relative to each other. And likewise, one can compare conditions 1 or 2 versus condition 4. And what one observes here is a value of about 0.43 and a spatial correlation of 0.91. Now, the reason I mention this example is so that people realize that a high correlation doesn't mean that they're identical. And one can detect very subtle changes in topographic distribution by looking at a measure like dissimilarity. So the other point that you should take away is that global dissimilarity is exclusively dependent on the spatial distribution of the data and not their strength, because you've already normalized the data. Hopefully that's clear to everyone. I see a number of chat comments. Let's see if I can look at some of these, because this is where people start to usually ask questions.
I'm receiving the questions, Micah, so please go ahead. I can let you know during the Q&A.
Okay. All right. So again, dissimilarity is exclusively telling you if I have a change in the topography, independent of the strength. And that can be useful because you want to look at these two measurements independently of one another. To look at the statistics on global field power is straightforward. It's like any other waveform. So if you're used to analyzing voltage waveforms, you can look at global field power, which is indeed a voltage measurement. So this is microvolts as a function of time, and you can look at this statistically in any way that you're accustomed to. It's interpretable as a modulation in the strength of the response. Now, the strength of the response can modulate either because it's more synchronous activity, more specific activity, less specific activity, and I can't draw direct mechanistic inference about that, but I know that it's a stronger versus weaker response. Global dissimilarity, on the other hand, requires more complicated statistical analysis because you're taking one measurement of the difference between two electric fields, the scalp, either in time or across conditions or across populations. The simplest, of course, is when you only have two conditions. So I'll use that today as an example, just to make things quite clear for people. So you have, of course, your observed dissimilarity, and if you permutate the data, you can start getting different values of dissimilarity for a given permutation. This is a Monte Carlo or bootstrapping approach, and this allows you to get an actual distribution of values and to determine where your observed value lies in that distribution. Allows you to get a p-value. And you can, of course, do this not only for one moment in time, but you can do this as a function of time. So you know when in time your two conditions, in this particular example, differ topographically. Now, what you don't know is whether or not, and you can model that, let's say, by letters. So you have topography A for condition 1, topography B for condition 2, and a nice statistical outcome that you can just stop right there and say, great, the topographies differ, different networks in the brain are active at whatever latency you're looking at. But you don't know whether or not it's because there's one topography for condition 1 and a different topography for condition 2, or another scenario, which would give the same statistical outcome. You have one topography for condition 1, but condition 2 goes from topography B to topography C. You can also then imagine another scenario where condition 1 goes from A to B, and condition 2 goes from B to C, and so on and so forth. all of these different possibilities make the fine level interpretation of a topographic difference more complicated. It's already one thing to be able to say with confidence the topographies differ. It's another thing to be able to say this is how they differ. And this is a real challenge. So on to the next full question. Latency and polarity define an ERP component. True, false, or it's like porn. I know it when I see it. I should add that that quote is from a Chief Justice of the United States, and it's not my own quote. Okay, hopefully everybody's had a chance to vote.
Interesting. So 64% true, 28% false, and the rest option 3.
Fair enough. Again, glad to see everybody's got their confinement sense of humor still going. Right. Nice. So the quick answer to this is false. A component is defined by its topography and its latency, not by its polarity. And what I'll do is I'm just going to jump out of this just to make this point very clear. Where was it? So if we look at this image here, right? If I only use the polarity, if I say that this component here has to be negative, but I change the reference, it's now positive. And you can also see that in the maps. The color that I ascribe, whether it's above or below 0, can change as the choice of the reference. However, the shape, the distribution does not. So again, that's an important feature of topography, is what should be defining components, not their polarity. And if we come back, oops, here. So given that we want to define components by their topography, how do we do that? How do we sort out the alphabet soup? How do we cluster the data to know what these letters are so that we can better understand when we have a change in topography across conditions, groups, etc. And if we can do that, then we have a way to identify components in a completely unsupervised and data-driven manner. There are, of course, other approaches out there, people that have surely heard of these, whether they be principal component analysis, independent component analysis, so on and so forth. What I particularly like are methods that are unsupervised and data-driven when looking at the data. Cleaning the data is a different issue in terms of being able to remove components linked to artifacts. So if you were to look at a time series of event-related potentials or even resting EEG, what your naked eye can hopefully observe is that the topography doesn't randomly fluctuate in time. There's structure to this data, and it's possible to then cluster the data. And to cluster, in this sense, means to reduce the data to a representative set of topographies that explain the maximal amount of variance in the data set. And this can be done, generally speaking, there are two families of data-driven approaches for topographic clustering. One is k-means, and we'll get to that in a moment. The other is hierarchical clustering, and then you can have variations upon that, one of which is atomized and agglomerate hierarchical clustering. The main difference between these two large families of approaches is that k-means operates independently for each set of n template maps, and I'll explain that in just a moment. And therefore, the results can vary slightly between applications to the same data set, and it's particularly useful when you have data with very little variation in global field power. The hierarchical approaches operate interdependently as you go from n to n-1 template maps. It doesn't vary between applications to the same data set because it's completely based on global explained variance. And it's particularly useful in our experience when applied to event-related potentials. So in a nutshell, what hierarchical clustering does is it plays the game of you're the weakest link. And it takes the data set and eliminates that moment in your data set that is explaining the least amount of explained variance and relabels it according to another cluster. So I don't have time to go through all of the families of clustering today. I'm happy to address questions about them. But let me just give you a feel for what k-means clustering does, because I think it will serve people well to understand that this isn't some magic algorithm. So again, imagine you took your data set and just threw it in a bucket, and you stripped off labels of condition and time and even group, and really literally put all of your data in a bucket. So each map is one instant in time, but we don't know which instant in time, we don't know which condition it came from, nor what group. We have all that information, we've just put it aside for the moment. What I'm then going to do in a k-means clustering is I'm going to say, I think there are 3 maps or 3 clusters that would describe my data set. And what I'm going to do is for each and every data point, I'm going to calculate the spatial correlation between each of these 3 maps and each data point. And from that, I can say how much of the global variance is explained by these 3 maps. And there might be periods or data points where the red map has a highest spatial correlation, others where blue, red, green, so on and so forth. I can then take an average of all the data points where the red had the highest spatial correlation and get a better red, if you will. So I'll take an average of all those, make a new template map, and then have a better red and better blue and a better green. And I can repeat this process of going back to the original data and calculating the spatial correlation. And I can do that over and over again until the global explained variance doesn't change. And what that tells me is this is as good as 3 template maps will be in describing these data, or these 3 template maps. Now, because I took 3 data points out randomly, it's quite possible that I took neighboring points that are already themselves highly related to each other or highly spatially correlated with each other. So I picked randomly very poorly. I didn't do a good randomization. So I have to repeat this same thing over and over again, taking n template maps, let's say 3, until I get the 3 that explain the most amount of the explained variance from my entire data set. So that's 3 maps. Now I have to do the same thing for 4 maps, 5 maps, 6 maps, 7 maps, etc. And I can do this over and over again, and now I have the best 3, the best 4, the best 5 template maps that describe my entire data set. So now I have 2 challenges though. How many is enough? And what is the pattern observed back in time, condition, group, etc.? Well, the second point here, we never lost that information. We stripped it off for the clustering, but we kept it. We could always put those labels back on. The question of how many maps best account for the data is always a trade-off. On the one hand, you can have as many maps as you have data points, and that explains all of the variance, but you haven't compressed the data at all. On the other hand, you can go the other extreme and have 1 map that explains the data set, which will probably compress the data very well, but it won't describe a lot of the variance in the data. So somewhere in between. And there are many, many approaches for this. One of which I illustrate here, which is based on notions of dispersion, which we've implemented in some of our analyses. The basic take-home message, again, I'm trying to target this to newcomers, is what you want are clusters that are wide apart from each other and have very little variance around their centers. And when you do that, you've done a good job in clustering your data. Practically, what that means is you can go back and relabel your conditions or groups as a function of time with different maps. It could be blue. You can use colors, you can use letters, you can use whatever moniker you want to use, but it gives you a hypothesis at, in this case, the group average level for what's happening, let's say, in condition 1 versus condition 2. So you might have the hypothesis that condition 1 has a yellow map and condition 2 an orange map over this time window. And what you can then ask is, for each and every participant, how well does the yellow map observed at the group average level describe the data from the individual level versus the orange map over that same time window for condition 1, and likewise the same thing for condition 2. And you can quantify these pie charts as the frequency of map presence and perform, in this case in ANOVA, comparing map by condition. And the interaction between these would tell you that one map is better characterizing responses to one condition and another map to another condition, which you can see in the histograms here. Now, that's one type of dependent measure that you can take from topographic clustering. You can also take measures like global explained variance. You can look at the first onset, last offset. You can look at Markov chains to look at the sequence of maps in time. There are any number of different dependent measures you can take once you have a good topographic clustering of your data set. So, final poll question. Signal averaging is required to observe an event-related potential. True, false, or still like foreign? I know it when I see it. Okay, hopefully everybody's still hanging in there and asking themselves some fun questions.
What is the reaction to the final poll? I'd like to close the poll now. Well, let's share the results. True, 50%, false, 46%, and still like porn, I know it when I see it, 4%. Thank you for your participation.
Thank you, everybody. So you guys seem a bit on the fence with this one, and the simple answer to this is false. It's not required to observe event-related potentials to signal average. It can be observed in single trial data. And you can see this in methods that are based on single trial topographic clustering. Again, what one can do is model the data as a mixture of Gaussians, clouds of data points, and ascribe to each and every trial a color, just like you saw before in the clustering of average event-related potentials. Here it's single trial, and what you'll see are bands of color in time. And if you don't believe this, you can also go to more well-known applications of single trial EEG analysis. Brain-computer interfaces are entirely based on single trial signal analysis. Same thing with MVPA, or multivariate pattern analysis, which has become very fashionable under the moniker of representational similarity analysis. This is all based on single trial event-related potential analyses. So what you should take home from this is that indeed, looking at trial averaged event-related potentials is of course one way of looking at your data. But by looking at topographic information, in fact, the signal to noise is quite increased. In time, it's very different. Typically, we'll look at one electrode and a time series, and it's hard to see the signal in that with the naked eye. But looking at single trial topographic information, one can often see the signal quite well at the single trial and single moment in time level. So again, just to close the circle a bit, EEG's come a long way since the 1920s in terms of not only how we measure it, in terms of the number of channels, the kinds of computer and hardware equipment behind it, but also in terms of the environments that we can record from, the types of scenarios that we can conduct our research in, the types of populations that we can approach, as well as the inferences and interpretations I think that we can make mechanistically. So what you should hopefully take home is that global measures are reference independent. They benefit from high density EEG montages. These measures are mathematically and statistically straightforward. They're readily implemented and readily interpreted. You can get around or circumvent a lot of the guesswork and ambiguity that a newcomer to EEG might confront. And I think these global measures allow for an easier multicentric approach to our research. And this is something very important nowadays in neuroscience and clinical research, is being able to replicate across labs, across studies. If you're using global and reference-free measures, this is entirely facilitated. So EEG has been a workhorse in neuroscience for over a century, or nearly a century, excuse me, that has provided us with brain correlates and constructs in experimental psychology. It's also a true neuroimaging technique. It can be translated and it can be applied not only in animal models, but also in computational and in silico models. This versatility and accessibility in combination, I think, with signal processing advances allow for this old dog to still deliver new tricks and innovations. So I'll stop there. I know I've gone a bit long, but hopefully it's stimulated you to do some thinking and to come up with some tough questions for me.